In this video, we are going to discuss a topic: what are gene cloning vectors? A cloning vector is any DNA molecule that has the ability to replicate inside the host to which the desired gene has integrated for cloning. Cloning vectors include plasmids, bacteriophages, lambda-phage vectors, M13 vectors, cosmids, BAC, YAC, shuttle vectors, etc. Now let us move into the details of a cloning vector. Let us take an example, an ideal vector, that is PBR322 plasmid Bolivar and Rodrigues 322. The three essential characters for a vector are an origin of replication, you can see right here, then many unique restriction sites within selectable marker regions, and at least two selectable markers. Now let us move into the detail. Why we need an ORI, origin of replication? So usually our host is bacterium. You can see the genomic DNA, bacterial DNA right here. So we are introducing our vector by transformation. So an ORI, origin of replication, are sequences that are required for replication of this vector inside the host. Now the second part, that is the unique restriction sites. In PBR322 vector, there are 4 unique restriction sites: BAM1, SAL1, then PVU1 and PST1. What is it for? So let us take these as an example. Here we are going to introduce our gene of interest into the BAM-H1 restriction site. So we are making a cut using BAM-H1 restriction enzyme, and we are introducing our gene of interest there. So restriction sites are sequences where specific restriction enzymes can make a cut so that our gene of interest can be incorporated into the vector. Now the third thing, that is the selectable markers. In PBR322 there are 2 selectable markers. You can see right here, this is a tetracycline resistance region. This region has genes responsible for providing them with resistant to antibiotic tetracycline. So you can see the second marker, that is ampicillin resistant region. This region has genes providing resistance against ampicillin. A host carrying this vector can grow in ampicillin containing medium. Now, after transformation, we will be getting 3 types of colonies. Non-transformed, without our vector, majority of the colonies will be non-transformed without our vector. Then transformed with non-recombinant vector. Our gene of insert may not be integrated into the vector. And the third class, that is transformed with recombinant vector, or transformed, the one which is having our gene of interest incorporated. Now how we select our recombinant ones from all these colonies? Here comes the use of selectable marker. So now we know that non-transformed one cannot grow on ampicillin or tetracycline medium, as that host or bacteria doesn't have ampicillin or tetracycline resistance genes. So we are simply plating that into an ampicillin or tetracycline medium so that colonies cannot grow. Now the second category, that is transformed with non-recombinant vector. Transformed ones with non-recombinant vectors have both ampicillin and tetracycline region so that it can grow in both ampicillin and tetracycline containing medium. So it will form colonies right here. Then the third category, third type of colonies, that is transformed with recombinant vector. Our gene of interest is inserted. So we need to pick out these colonies. So growing such colonies on ampicillin or tetracycline medium, the result is, in this example we have incorporated our gene of interest in PAMH1 tetracycline region site. So transformed with recombinant vector carrying our gene of interest in this. So if the gene insert has integrated properly, the tetracycline region gene is now malfunctioning, often referred as insertional inactivation. Thus, our transformed recombinants can grow only in ampicillin medium and cannot grow on tetracycline medium due to insertional inactivation. So, recombinant colonies can be easily selected from the master plate. By comparing these, recombinant colonies can be selected. Hope everything is clear.